Well, this is the last of our three-part series on translation. What I'm going to do here is I'm going to cover a couple of the differences between what we find in prokaryotes and eukaryotes. And then I'm going to talk a little bit about how proteins are modified after they're made. Particularly, we're going to focus on those proteins that are made within the ER and then are exported from the cell or end up on the cell membrane at the end of their processing. So these are the three parts that I want to cover in contrast between prokaryotes and eukaryotes. And one thing to remember is that prokaryotes don't have a nucleus. And so that kind of makes them a little bit special in the case of transcription and translation because both of these things are occurring within the cytoplasm. And what's pretty cool about this is that it allows for bacteria to not only transcribe an mRNA, but translate it at the exact same time. And remember that prokaryotes, their RNA doesn't need to undergo any sort of modification like we find in eukaryotes. And so what we can find sometimes here is if this is our mRNA, we might find that it's still being transcribed, but we've also got multiple ribosomes bound at one time here, making the same protein again and again and again, and these structures are called polysomes. So this is our protein coming off of each of these ribosomes. We see these polysomes also in eukaryotes, but what's different is that the transcription has already occurred and the mRNA has been modified before translation is occurring on it. Now, in the case of eukaryotes, again, transcription is going to occur in the nucleus. That's where we're going to see modification of the mRNA from that precursor mRNA that we talked about, and then translation is going to occur in the cytoplasm. And keep in mind here that this translation could occur on free-floating ribosomes that are just within the cytoplasm or that liquid itself, or they could be ribosomes that are docked up onto rough endoplasmic reticulum or rough ER. And that's what we'll talk about in the next slide. Finally, one difference here that we find between prokaryotes and eukaryotes is that first AUG in eukaryotes is typically going to be the first one that occurs somewhere after that 5' cap. Also, what we're going to find is that the ribosome actually scans for what's known as a Kozak sequence. This sequence is just a short strand of nucleotides that basically signifies to the mRNA or to the ribosome that the next codon or the next AUG after the Kozak sequence is going to be the start codon. Prokaryotes have something similar, but theirs is called a Shine-Dalgarno sequence. And so again, the importance of these two sequences here are just to signify to the ribosome that the next AUG is going to be the start codon for that translation. Now here's a picture of a eukaryotic cell. So in purple here we've got our nucleus. In green we have our rough ER studded with ribosomes. You can see there's some free-floating ribosomes in the cytoplasm as well. And then we have our Golgi apparatus in orange here. And what we're going to talk about very, very briefly is how mRNA can be translated on rough ER and the modifications that it undergoes. Recall that for most free-floating proteins, ones that would be found, for example, in the cytoplasm, these are going to be ones that are made on these ribosomes that are kind of free-floating in the cytoplasm. However, proteins that have to be excreted from the cell, so ones like insulin that are going to be moved through the endocrine system, those are going to be made on rough ER, and particularly they're going to be made inside of the rough ER. There may be some membrane-bound proteins too that we find made on the surface of the rough ER. After the initial making of these proteins, they will get transported to the Golgi, where they undergo post-translational modification. This might occur, a couple of things that might occur in this process might be cutting of the protein or removal of what's known as a signal peptide, which helps to designate where that protein is going to go after the modification. Additional modifications might be the addition of lipids or sugars that would be used to help anchor the protein in the cell membrane, for example. So like what we see with RAS and some of those other G proteins that we talked about in the past with cell signaling. Or we might see additional modifications, as I mentioned, like sugars that get added to the surface of these proteins that might be found as cell recognition markers. And so here in light blue would represent maybe that sugar that's being added to this. So just to summarize what we've said here, translation, regardless of whether the protein is a membrane-bound protein, a secreted protein, or one that would be free-floating within the cytoplasm, they're all going to be made through the same process that we talked about in our previous video. However, where they are made and where they might be modified are going to be slightly different depending on where that protein ultimately is.